Thank you. So my name is Chen Wu, and here I post my Twitter if you're interested. So today I'm going to talk about my work titled Individual Differences in Autistic Traits are Associated with Reduced Goal Emulation in a Computational Model of Observational Learning. So observational learning is very common in our everyday life, and people have defined two basic strategies. One is called goal emulation, that means you infer the goals and intentions of other people. And the other is called action imitation, that means you copy the action of other people, but do not make any inference at all. And autism is characterized by a variety of difficulties, especially in social functioning, and it represents traits that are distributed across the whole population. And we know that autistic traits affect observational learning, but what is unknown is that one specific process during observational learning is specifically affected by autism. So in our experiment, I collect data into independent large-scale online samples, and then I administered my observational learning task that could dissociate emulation and imitation very clearly. Then also I collect the data of social responsiveness scale as a measure of autistic traits. I hypothesized that more autistic traits will lead to less emulation in observational learning. So here is my task. It is a quite complicated task, but basically the participants choose from one of the three slot machines on the screen, and they can get a token of either blue, red, or green, depending on the probability of these colors specified as proportional color in the slot machine. The participants themselves do not know which token is rewarded, but they could observe another partner playing the same game. So through their observation, they can figure out which is their choice. And we define two strategies. One is imitation. That means the participant just simply copy-paste the previous action, the previous slot machine chosen by the partner. The other strategy is called emulation, which is the participants figure out, they infer which token is the goal from the partner, and then they choose a corresponding slot machine based on their inferred token. And finally, we fit the behaviors from our participants using a set of computational models, and we cluster their behaviors into 4 groups. So we found one group who mainly used imitation strategy, another group who mainly used emulation strategy, another group that combines both strategies, which we call arbitration, and finally a group that does not learn anything actively at all. So we look at associations between autistic traits and the proportion of choices consistent with the emulation in the study, and we found that there's a negative correlation means that people with higher autistic traits perform less emulation in this task. And also, if we look at autistic trait level by the 4 strategy groups, we found that the autistic traits is highest in non-learning group, followed by imitation group, the emulation group, and finally the arbitration group. So basically, our results have confirmed our hypothesis. That's it for this research. I would like to thank my advisor, John O'Doherty and Ralph Adolphs, and also Caroline Charpentier, who has developed this amazing task and mentored me throughout this project. Thank you.
Awesome. Next up, Liberty Hamilton with the title for something that looks like it should be a long book or a 3-week course maybe, but
Yeah.
it's going to be a lightning time.
All right. Okay, hopefully the Zoom sees the, okay, looks good. All right, so just to preface, this is not actually my field, but this is something that I've thought about because of what we do at NeuroHack and when we're thinking about what we're doing with open data sets. So I want to talk a little bit about a paper that I recently learned about, and this was looking at machine learning for something kind of serious. So this was an fMRI study where they were trying to predict suicidal ideation based on 2 groups of people, 17 controls and 17 people who had been reported to be suicidal ideators. And this came out in 2017. And so this made a big splash. And so, it had a lot of attention. There's a lot of news articles about this. People are, talking about it. There's news media that's, can we use brain scans to predict suicidal tendencies? And so they say here, they use this naive Bayes algorithm, and they showed that the algorithm they developed was 91% accurate in identifying whether a person was from the suicidal or control group. So this is, and was accurate in identifying people who actually attempted suicide. So this is big and serious, and they thought may be important for being able to identify this risk. However, after this paper came out, there were some critics. Specifically, NeuroCritic, this is a blog that came out around that time. And this person started to say sort of some reasons why they thought that maybe these results were not as trustworthy as they should have been taken to be. And so then what happened after that is there was more press on how, okay, actually these MRI scans don't reveal suicidal thoughts. And there were several researchers also in neuroscience who posted first an archive preprint that was basically reanalyzing this data to show that this result was based on overfitting of data. And so really training and classifying on the same data sets. I should say that the actual task that they did was they had people in an fMRI scanner and they were just looking at words. And some of these words were emotional words and some of them were not. And then the brain response to these more emotional, either negative or positive valence words might predict this group. That's what they said. However, so Versailles and Cording first said, okay, we don't think that this is very trustworthy. And they also, this gets noted in some other papers as well. So this sets off a whole slew of people talking about how this is based on very few observations. Remember, there were only 17 people in each group. And usually for this type of thing, you really want hundreds of participants to be able to say something concrete. So they eventually published this year a paper in the same journal in Nature Human Behavior, where they show, using this same data set, they reanalyze it, and they show that actually these results that they had in their original paper were not robust, and the article is retracted. So this article was retracted 6 years after it was posted. But what I wanted to talk about a little bit is not to really cast any blame necessarily on the authors, but I do want folks to think about the ethics of these sorts of scientific questions. So if you are working with a data set and you want to do something like classify whether someone has a neuropsychiatric diagnosis, think about what is that going to mean? What is the potential for harm versus benefit? How rigorous do you need to be to really believe that this is a result that you should use? I think, yeah, so this is what I just said. I think thinking about the ethics of what kind of hacks we choose to do and what that means in terms of, I don't think it means that this stuff should never be published, but I think the onus is on the researcher to really make sure that what they're doing is repeatable and robust. So thinking about how to avoid both false positives and negatives, and which of those is more important. Mitigation of harm, and what is the potential for harm for any analysis that you're thinking about doing. And then thinking about how open data are used. So for a lot of data sets that we have, I work with patients. And so sometimes I'm not particularly interested in looking at their emotional state, but we have neuropsychiatric reports. We have things like the Hamilton Depression Scale. You could do some of these other analyses. But then my question as a group is, I think we should think about whether we should and how we should do those so that they are actually robust and repeatable and that they are benefiting rather than causing harm. So that's my soapbox.
Okay, next, Tanya to present something about not human. Let's share the screen.
Hi, so my name is Tanya Miramontes, and I'm going to talk to you about zebrafish. So tiny fish, big discoveries. Why we use zebrafish as an animal model, or why you can kind of think about them as why they're a good model. They're just like us, have a spinal cord, have a brain, have eyes, but also 70% of our genes we share with zebrafish. They have rapid external development, so you can see this video. This is from a 2-cell stage, and then at the end of the video is about 20 hours or so. You can see they start to look like a full organism. So I'll just let that play. Around this stage is usually they're born in the morning and you can see the stage by the end of the day, which is really nice. And then this happens like middle of the night, and then you could start to see up here, the head develop, the spinal cord and the tail. So it's really nice. And then they're small, not that expensive compared to using mice or rats and humans. You could fit hundreds in a tank, and you could have a rack of like thousands, and it won't be that expensive. They just run on water and like fish food. They're small, so this is what they look like around 4 or 5 days post-fertilization. This is when I use them for my experiments. They're about 3 millimeters long. We can have various transgenic models, different colors, cross them, and you can see they're also optically transparent. So I use them, and other people use them for in vivo imaging and neuroimaging. The imaging power you could see here. This is an oligodendrocyte precursor cell, and within the matter of a couple of hours, it matures and creates myelin sheath. And this is a single cell. The animal is alive. We didn't cut it up or do anything to it. And this is using confocal imaging. We could get this resolution using like the 20x or 40x objective. And then this is another one. It just, you can see how cells are different. That the top one in about 12 hours kind of is mature. This one is taking a little bit more time to kind of mature. And just lets us see the heterogeneity and the dynamics on an individual cell level. And then finally, you can use they're very good with genetic tools using CRISPR-Cas9. Could easily generate multiple stable mutant lines. You could also do genetic screenings. And something that people don't know is you could also do pharmacological screens, so they can absorb small molecules through the skin and test a bunch of different compounds, image them, and look at their readout at a cellular level as well. So yay fish.
Thanks a lot. Next up, Sebastian. Was something that initially when I read the title, I thought it might be unethical to do, but then I realized that it was nailing the subject.
I'm not gonna nail anyone, hopefully.
Don't forget to share your screen.
Does it work?
I think so. Okay, so I want to present an idea, a really powerful idea of synergy, which can help us to nail down explanatory biomarkers in the brain. And the motivation for this is we of course want to find links between brain states through neuroimaging into neurobiology, and we would like these biomarkers, these bridges between brain states and neurobiology to not only classify brain states as machine learning algorithms can be, but also be interpretable in terms of mechanisms. And so I want to show you now, switching to a little side story, an idea which might help us to do that. Let's imagine that one of these, what happened?
You're muted.
I'm muted. Okay.
Okay.
Let's imagine that one of these suspects has stolen some cookies, and we have multiple sources of information as in the brain. And one of the sources of information tells us that the suspect had a mustache and glasses. This allows us to identify, eliminate some of the suspects, but not all of them. Another witness gives us a different set of information that the suspect had a head and the glasses, which allows us to eliminate a different subset of the suspect, but still not everyone. And only the two sources combined allow us to eliminate enough suspects so that we can uniquely identify a culprit and put them to jail. And what this example shows is that not all information is the same. Some information is unique, but some information is also redundant between the two sources, and some information is synergistic between the two sources. So only the two sources combined can give us the ultimate information that we want, and neither of them separately. And this can be formalized into some mathematical frameworks and can be measured. And what it means in the brain is that if we have multiple sources of information, like a variable, like a brain region X and a brain region Y, and we want to predict the future of one of these brain regions, some information might come from one of the other brain regions, so from X to Y directly, but some information might be synergistic between the past of X and the past of Y into the future of Y. And when I apply this to brain data, in this paper, these authors use just a measure of this information flow, which combines all of these different components, and they find very different maps for all the incoming information into all brain regions on the left side and all the outgoing information from each brain region. And what I did is I decomposed this information into the synergistic and unique components, and lo and behold, the synergy dominates the brain interactions, while the unique direct transfer is not present. And this was done also in REST and in 7 tasks of the HCP project. And what this shows is that the brain seems to be more than the sum of its parts. And if we take out any of the parts, any of the circles, none of them are going to be connected, right? And well, the question is, does synergy break down in brain disorders, and can we use this as a potential biomarker? And that's, I suppose, for the future. Thank you, and of course, the effort is very synergistic itself.
Wonderful, thanks a lot. Next, yeah. Brain symmetry.
Do we do HTML people? Yes, I have a Zoom.
So to share screen.
Okay.
And then go ahead and present.
Okay. Okay, hi everyone. So today I'm going to talk about brain asymmetry and language lateralization, and just briefly talk about what I'm doing in my PhD. So brain asymmetry is a very, very hot topic, and you've probably seen a lot of people talking about people being left brain, right brain, and while this is very generalized way of thinking about the brain, some dimensions, brain asymmetry in some dimensions actually true. For example, in language, there's 90% of people that are, lateralized in the left hemisphere. Whereas 10% of individuals use both hemispheres or right hemisphere for language. So there is a variability, although majority of people localize their language on the left side of the brain. You would ask, why is it important to know? Well, in epilepsy cases, for example, we need to understand where the language is located, because when we get rid of a seizure, we don't want to get rid of language. So traditionally, people use task fMRI to localize the language function, but it's not ideal for certain populations. For example, when we have children or certain people who cannot stay long in a scanner, it can be problematic because task fMRI requires a long time, and it just can be problematic at some point. So our goal is to look at the structure of the brain and see whether we can find what hemisphere, or if it's both hemispheres, what hemisphere people use for language, to make sure we avoid those specific areas when we do epilepsy surgery. So in my research, what I did, I specifically looked at the white matter and so looked at whether there are any associations with task fMRI, language function, and structure of white matter. So I looked at the microstructure of white matter, which is also defined as quantitative anisotropy. So in simple terms, for people who don't really know much about diffusion MRI, it basically measures the integrity of the white matter. And I used, for that specific approach, I used connectometry, which basically finds a specific streamline. It doesn't matter, it's not constrained to the bundle. It can find a specific streamline that is significantly associated with left hemisphere lateralized bilateral or right hemisphere lateralized people. And then in my second approach, I use shape analysis, where I looked at volume, diameter, curl, elongation, and other parameters, and looked whether the symmetry, for example, between left arcuate fasciculus and right arcuate fasciculus is associated with the function of the language. So long story short, I found that microstructure of white matter, but not macrostructure, is associated with language lateralization. And in my study, I found that bilateral individuals, in other words, individuals who use both hemispheres for language function, are having higher white matter integrity in anterior commissural tracts. What does it mean? Well, a lot of literature previously used to kind of define bilateral individuals as being impaired, but we don't think this is correct because in our studies we used HCP data sets, so everyone was healthy. There might be that people use compensatory mechanisms to make sure that they transfer a neuronal signal from one hemisphere to another as efficiently as people who lateralize their function. But this is our hypothesis. So next step is to basically see whether bilateral individuals have their microstructure of corpus callosum correlated with, for example, IQ or cognitive scores. And since we use HCP data set, language function task was very specific to language comprehension. So we are now working with our collaborators to work on Bill Engine data set and see whether we can replicate our studies in different language tasks like language production and verbal fluency tasks and so on. And if you have any feedback or comments, I'm more than happy to chat with you. Thank you very much.
Okay, next up is Lester. Which is going to round us up with even more different kinds of methods of measurement.
Hi, everybody. So I'll be talking about source separation in PET imaging. So in PET, right, we can label or we can find different parts or pieces of the neurotransmitter systems by using radio tracers. And in my case, I'm working with a tracer called PHNO that allows us to identify dopamine D2 and D3 receptors. So we can pick out different dopaminergic regions of the brain, like the substantia nigra, the globus pallidus, or the ventral striatum. The problem is that this tracer exhibits affinity for two types of receptors. So if you are interested in just viewing one, we can't do it unless you figure out a way to disentangle the signal. So in the hypothalamus, most of our signal is going to be due to D3 binding, whereas in the dorsal putamen, most of our signal will be due to D2 binding, and it's always going to be a weighted sum. Now, potentially, and this is what my lab was doing, we can use an algorithm called independent component analysis to separate out those signals. This is frequently used in functional MRI data to identify functional networks by grouping together voxels that share common variants. In the case of PET, we are grouping together voxels that share common variants in their binding potential values across space and across subjects to identify source networks in this case. So for those who are not familiar with ICA, I can do a simple analogy. If we record a conversation at a party, this algorithm can figure out how to piece apart that recording and determine, well, this person is saying this person is saying this, and this person is saying this. And it can tell us how relevant each of those sources is to our signal. If we do it on PET data, it's exactly the same thing. We can piece apart the sources of our PET data, where it's coming from. My lab did this to identify two specific sources in this PHNO data, a striatopallidoid source that was specific to D2 receptors and a pallidonigral source specific to D3 receptors. And the importance here is that they found that if they look at healthy controls and patient participants with cocaine use disorder, that the changes are different. There was a decrease in this D2 source intensity, but there was an increase in D3 intensity with cocaine use. So where I'm applying it is on this data set where we had PET scans, pre and post amphetamine administration. So we used amphetamine as an acute inducer of a state where dopamine is going to be more active and displacing our tracer. So we expect to see a decrease in signal. Now the question is that different between smokers and non-smokers? And so what we found, or what I found, was that in this striatopallidoid source, this D2 receptor specific source, we have a session effect, meaning that after the amphetamine, dopamine goes up, the signal goes down because it's displacing our tracer. But in the non-smokers, the decrease was steeper. So if you see the graph on the top right, there's an interaction effect here. The non-smokers do not have a blunted dopamine system, so amphetamine is able to induce more dopamine, which is able to displace the tracer more. Whereas the smokers do have a blunted dopamine system, and we were able to pick that up with the ICA. And with that, I'll acknowledge my lab, and thanks.
Yes, next up. Comment.
Let me share my screen real quick. Thank you. Okay, hi everyone, I'm Gafkar. I'm soon to be a second year PhD student in human development at the University of Maryland, working in the lead lab. So I figured I was one of the few who works with kiddos. So I would just like to tell you guys how it is to do neuroimaging with children. Joys and challenges of it. This is our team. Let me make this smaller. And we do neuroimaging with kids. Yes, so just a few considerations when imaging kids. So you usually get shorter data collection time per visit. So for example, our MRI, the entire sequence of MRI visit lasts no more than 20 minutes inside the scanner. And if we are very, very lucky, if we get those 20 minutes before the child is like, I'm done, I'm getting out of the scanner. And then for the FNIRS collection is the same thing, 20, 30 minutes per visit. We just had a kid who in the middle of the task was just like, I'm done. And just like, that's it, going home. So you have to account for that when doing neuroimaging with kids. Another thing is also we have to train the kids in the mock scanner before bringing them to the real scanner. So we have this replica of the real scanner at the center where we invite the kids to go in and then they get to watch their favorite movie. And then we pause the movie. If they move, we'd be like, stay still. If you want to watch a movie, you have to stay still. And we also walk them through what it is, a scanner. We tell them it's a brain camera. So if you want to get good pictures of the camera, you have to, not blurry, you have to stay still. And then familiarize them with the with the scanner noises so they don't get scared when they're inside the real scanner. And we take them to the real scanner after that. Another thing to consider is just the data processing. So you just have to be ready to get the very more motiony data compared to if you're just scanning adults. And this is normal for working with the kiddos. Data attrition is another thing to consider. So after all the cleanup is done, clean the data, quality check, we just have to be ready to lose more than 1/3 of the data collected for many reasons. Children sometimes just don't come to the MRI visit, or because of the too much motion going on, we have to cut and all of that. But and kids are just unpredictable. All sorts of things can happen when they are inside the scanner. So I'm not going to go over this really in detail, but just quickly, 2 big projects that are going on in the lab. The first one is called Family Inspired Neural Synchronous Study, or FINS, where we get to go to the preschool centers or child centers, and we scan with the FNIRS, which caregiver is the child, while they're wearing the 22-channel montage for the headband on the front. And they get to play puzzles, do emotional tasks. And this study specifically looks into how synchronous caregiver-child interaction predicts better social and cognitive outcomes. Another study that I'm more involved in, it's called PLANES, Preschool Language and Neuroengagement Study, where it's a longitudinal study that looks into how early experiences can predict brain development and language development. And so we do 3 visits at time 1 and time 2. We collect the behavioral data. Then we also get to ask the families to collect all the auditory environment that children hear throughout the visit. We do FNIRS, and then with the 3rd visit, we do MRI. I'm happy to chat, just neuroimaging with kids or about these specific 2 studies. But yeah, thank you. Thank you.
Very cool.
What is this? So usually I do a portion so that I can see it.
You showed us a portion of it. That works.
Cool. Um, Okay, cool. Hi everyone, I'm Anna. I'm here to tell you a little about some work I've been doing using virtual reality to collect behavioral data. So as humans, we've got this limited field of view, can't see anything behind you, but we still need to operate in a 360-degree environment. So the idea of the study I'm going to tell you about is that perhaps in familiar environments, we reconcile this limited field of view by predicting upcoming scene views as we move our eyes and heads and bodies around in space. So there's a lot of previous work showing that making predictions about upcoming visual stimuli can improve perception of those stimuli, but how this works in naturalistic context isn't well understood. So in this study, we asked broadly whether memory for a 360-degree environment facilitates perception of upcoming scene views. And to find out, over the course of 2 days, we had subjects study a set of 18 real-world 360-degree environments taken from around Dartmouth's campus in virtual reality headsets, and their instructions were just look around like you normally would. So here's an unraveled view of one of the environments. Each one was selected to have an open view, like a field or a parking lot, and a closed view, like a wall or a facade of a building. The open and closed views appear on opposite poles of each scene, so relative to your starting position, you need to turn roughly 90 degrees to see an open view and roughly 90 degrees to see a closed view. So once participants are familiar with these places, we have them do a priming task in VR. So on each trial, they first see a prime image presented directly ahead of them for 300 milliseconds. And then after that, they turn left or right to make a perceptual judgment on a target view from a studied scene. So most of the time, the prime and target are adjacent views from the same scene. And the idea is that if predictions of upcoming scene views are generated across head turns, participants should be faster at making this perceptual judgment, this open-close judgment, if they're primed with a view from the same scene compared to a view from a neutral view of a blank gray rectangle, which shouldn't give participants any useful information about what they're going to see when they turn toward the target, or if the prime is taken from a different place and could therefore prime irrelevant information about the upcoming scene view. So I'm going to plot the results of this study here with reaction time on the y-axis. Using response time to the neutral condition of the baseline, you can see that, as expected, there is a reaction time advantage for same scene trials, which suggests that seeing a view from a familiar place primes perceptual judgments of upcoming scene views across head turns. Also, as expected, we find a disadvantage for different scene trials, which suggests that irrelevant information is primed on those trials. And to be confident that this effect hinged on subjects' memory for the studied scenes rather than on some visual confound, we ran the same experiment again, but this time using a set of unfamiliar and unstudied places. As you can imagine, seeing a scene view from an unfamiliar place wouldn't give you much information about what's coming next, so there should be no reaction time differences between these priming conditions. And that's exactly what we find. So when subjects aren't familiar with the primes, they show no difference in reaction time across conditions. So we think taken together that this work indicates that perceptual judgments of upcoming scene views are primed across head turns during naturalistic scene perception, and suggests that we may be generating upcoming predictions of upcoming scene views as we move our eyes and heads and bodies around in space. So to wrap up, I'd like to acknowledge all of the members of the Robertson Lab, especially my advisor, Caroline, and you can learn more about this work at this QR code. Thanks.
Thank you so much. And my last speaker for this session is Eric.
Hello everyone, I'm just share my screen.
I must also share that I'm super nervous right now because everyone's been doing so good. The bar is really high. But now we got it. Okay, so I'm going to show you my current work. I'm just starting my PhD, so this is still like something that came from my master. But I'm currently like drafting the manuscript for this work. And the title of the work is The Control Cost of Human Brain Dynamics. And just to lay the introduction, so I mean, basically the gist of this project is to quantify like how, or like investigate how the brain regulates itself, and in order to be as dynamic and athletic as this person here, and also how it pays the bill in order to sustain these control mechanisms. So I will start off by just showing you how network control theory works, but this is just going to be a very basic introduction. In case you have questions about it, you're welcome to come to me and chat about it. But the gist of network control theory is that we have a set of states that the brain goes throughout time. And the main output of using network control theory is that we can quantify what the control costs of these transitions are across time. And if you think about networks, you can think in the functional MRI world as resting state networks being the state that the brain fluctuates throughout time. So if we find a way to quantify resting state networks and then find a mapping onto time such that we can track how the brain is going from one resting state to another, we can actually go throughout time and quantify how much a transition costs to go from time point 0 to time point 9 and track all this control costs across time. Now, the final step of this is just to average across time points such that we can know what are the regions that are mostly engaged during resting state brain dynamics. And so as you can see, this map here is spatially very heterogeneous. So it has some regions that are participating more than others. And if you compare this to a map of oxygen consumption, it is also regionally very distinct. So some regions have a higher consumption of oxygen. And what is impressive here is that actually this map of regional control cost is actually associated with the regions that are mostly consuming oxygen, which means that the bill of how the brain regulates itself is paid potentially using oxygen as an energy substrate. So now the final, this is my favorite result, is this hierarchical rule. Now, instead of thinking of resting state networks, we can think of hierarchies. So we have, on the one hand, we have sensory, motor, and visual networks, who are unimodal networks in the hierarchy against heteromodal integrative networks, such as the other ones. And so if we think of hierarchies, then we can play through the game and track how often the brain is going from hierarchy level to hierarchy level. And it turns out that the brain is mostly confined in its own hierarchy most of the times, and rarely switches between hierarchies. And that can be actually explained through control theory, because it turns out that the transitions between hierarchies are actually more demanding than the transitions within a hierarchy, which explains how the number of times a certain hierarchy was visited can be predicted by its control energy. And yeah, this summarizes basically all of my work. Again, the title is Control Costs of Human Brain Dynamics, and my name is Eric Svartus. Thank you.
Wonderful. Thank you everyone for that great series of talks. Let's thank all the speakers again.
I also still have a question. Sorry, I have a joke. Why is Pavlov's hair so soft? Because of classical conditioning.
Thank you for that.